In molecular biology, housekeeping genes are typically constitutive genes that are required for the maintenance of basic cellular function, and are expressed in all cells of an organism under normal and patho-physiological conditions. Although some housekeeping genes are expressed at relatively constant levels in most non-pathological situations, other housekeeping genes may vary depending on experimental conditions. In a study involving cardiac stem cells, ACTB and GAPDH were found to be the most consistent, while I2M, HPAT1, and RPLP1 varied significantly between neonatal and adult cardiac cells. The origin of the term housekeeping gene remains obscure. Literature from 1976 used the term to describe specifically tRNA and rRNA. Interpreting gene expression data can be problematic, with most human genes registering 5 to 10 copies per cell. Housekeeping genes are expressed in at least 25 copies per cell and sometimes number in the thousands. Other commonly used housekeeping genes are GUSB, LDHA, NONO, 1 Papua New Guinean Kina, PPIH, RPLPO, and TFRC. Common housekeeping genes in human gene expression, transcription factors. ATF4 activating transcription factor 4, BTF3, E2F4, ERH enhancer of rudimentary homologue of Drosophila, HMGB1 high mobility group box binds DNA, ILF2, IER2 formerly ETO101 immediate early protein, JUND, LTBP4, LMO1 rhombotin 1, MYC also considered an oncogene when mutated regulates transcription of 15% of all genes, PAX8, PHF1 plant homology domain, PTTG1IP, SREBF1 sterile regulatory element binding protein Smith genus, TBP also known as Tata binding protein or GTF2D or TFIID, TCEB2 alungin, TCOF1 interacts with UBF to translate rRNA, repressus, ANF91, HPF7, HTF10 cruppel-associated box domain, ID3 inhibits DNA binding, PUF60, NF-kBIA suppresses NF-kappa, RING1, RNA splicing. ADAR, BAT1, FBL fibrillary, HNRPD, HNRPK, PTBP1, PABPN1, SFRS3, SFRS9, SFNRPA, SNRPB, SNRPD, SNRPD2, SNRPG, translation factors, EIF1 also known as SUI1, EIF3C formerly EIF3S8, EIF3D formerly EIF3S4, EIF3F formerly EIF3S5, EIF3I formerly EIF3S2, EIF3G formerly EIF3S7, EIF4A2, EIF4G2, TUFM to translational elongation factor mitochondrial, tRNA synthetases, IARS, KARS, RARS, ZARS, VARS2, YARS, RNA binding protein, PABPC1 poly-A binding protein, ELAVL3, ribosomal proteins, RPL3, RPL5, RPL8, RPL9, RPL10, RPL11, RPL12, RPL13, RPL13A introns contain genes for small nucleolar RNAs U32, U33, U34, and U35, RPL14, RPL15, RPL17, RPL18, RPL19, RPL25, RPL27, RPL29, RPL30, RPL32, RPL34, RPL35, RPL36, RPL37, RPL38, RPL40 transcribed with ubiquitin, RPLPO, RPLP1, RPLP2, RPS2, RPS5, RPS9, RPS10, RPS11, RPS12, RPS13, RPS14, RPS15, RPS16, RPS18, RPS19, RPS20, RPS23, RPS24, RPS25, RPS26, RPS27 transcribed with ubiquitin, RPS28, RPS30 transcribed with ubiquitin as the FAU, RPN1 riboforin anchors the ribosome to rough endoplasmic reticulum, MRPL9, RNA polymerase, POLR2A, POLR2E, POLR2J, POLR2L, protein processing, cyclophosphamidylserine 309 phosphatase inhibitor involved in protein folding, PPIH cyclophosphamidylserine HCANX folding of glycoproteins within endoplasmic reticulum, CAPNS1 calpain. Protease, NACA nascent polypeptide-associated complex alpha polypeptide, PFDN1 prefoldin 1, PFDN5 prefoldin 5, SNX3 sorting nexin, SNX17, SNX27, SSA2 protein translocation in ER, SUMO2 protein targeting, heat shock proteins, HSPA8, HSP90AB1, HSBP1, histone, CDH4 nucleosome remodeling, HIST1H2BC, H2AFY histone 2 subfamily, H2AFZ essential for embryogenesis, H3F3AH3 histone family, cell cycle, ARHGAP1, ACTDIA, CENPB centromere protein B, CTBP1, cyclophilin, CCND3, GAS1 blocks entry into S phase, PPP2. CB negative regulator of growth and cell division, PPP2 or 1A negative regulator of growth and cell division, RAD9, KIAA0174 or IST1 homolog, apoptosis, DAD1 defender against cell death, DAP, DAXX death-associated protein 6, oncogenes, ARAF, MAZ, MYC also considered a transcription factor, SAFB, RAB1A, DNA repair replication, COO80 also known as XACC5, MCM3AP possibly a primus, metabolism, PRKAG1 senses energy level and inactivates HMG-CoA reductase and acetyl-CoA carboxylase, carbohydrate metabolism, PRKCSH glucosidase 2, subunit beta, B4-GALT2, GUSB beta. Glucuronidase sly syndrome, chitinase, phosphogluconate dehydrogenase, GSK3A, hexokinase rate limiting step. No reference, H6PD, glucose 6-phosphate isomerase, phosphofructokinase 1, aldolase A aldolase C, triose phosphate isomerase, GAPDH, 1 Papua New Guinean kinase phosphoglycerate kinase, phosphoglycerate mutase, enolase, PKM2 pyruvate kinase, muscle, lactate dehydrogenase, LDHA lactate dehydrogenase, LDHB lactate dehydrogenase, TALDO1 transaldolase in pentose shunt, transcriptase, TSTA3 mannose metabolism, citric acid cycle, SDHA succinate dehydrogenase subunit A, MDH1 malate dehydrogenase, lipid metabolism, GM2A not highly expressed constitutively, HADHA trifunctional protein subunit alpha, HADHB trifunctional protein subunit beta, amino acid metabolism, COMT catechol-O-methyl transferase, GGTLA1 gamma-glutamyl transferase like 1, PHGDH phosphoglycerate. Dehydrogenase, ODC1, TPMT, nucleotide synthesis, IMPDH2 guanine nucleotide biosynthesis, NADH dehydrogenase, NDUFA1, NDUFA2, NDUFA7, NDUFB7, NDUFC1, NDUFS5, NDUFB2, DIA1, cytochrome C oxidase. Note that COX1, COX2, and COX3 are mitochondrially encoded, COX4A1, COX5A, COX5B, COX6A1, COX6B1, COX7A2L, COX7C, COX8, CYC1, UQCRC1, UQCRH, UQCRQC cytochrome C1, ATPase, mitochondrial, ATP5A1, ATP5D, ATP5G1, ATP5G3, ATP5H, ATP5I, ATP5J2, ATP5O, lysosomal, ATP6IP1, ATP6VOB, ATP6VOC, ATP6V1E, ATP6V1F, ATP6V1G1, lysosome, CTSD can degrade insulin in hepatocytes. Cystatin B may protect cell from leaking lysosomes. LAMP1, mannose 6-phosphate receptor, SGSH mutations can cause Sanfilippo syndrome. Proteasome, PSMB1, PSMB2, PSMB4, PSMB6, PSMA7, PSMB7, PSMD8, PSMD11, PSME2, UBA1, ubiquitin B UBE2I, UBE2, D2, UBE2M, USP11, ribonuclease, RNH ribonuclease inhibitor, oxidase reductase, TXN thioreductase, structural, cytoskeletal, actinin alpha-4, beta-actin, ACTG1 gamma-actin, ADD1, ANXA6, ARPC4-actin-related protein 2/3 complex, CAPZB, COL6A1, DNCL1 cytoplasmic dynein, EZRES-RIN, KIFC3 kinesin, mucin, MYH9 myosin chain 9, RHOA also implicated in regulation of cell cycle, September 1 homologous to CDC10 in SRVZ, September 2, SLC9A3 links SLC9A3 to cytoskeleton, TAGLN, TAGLN2, TMSB10 thymosin beta-10, TUBB-tubulin, beta polypeptide, TUBB4, WDO1 actin disassembly, organelle synthesis, a specialized form of cell signaling, BLOC1S1, April 2 M1, ANXA2, ANXA6, ANXA7, April 1 B1 coated vesicles, CLTA clathrin A CLTB clathrin BGP2 enzymatic secretory granules, KDELR1 Lys-asp glue tag that targets to endoplasmic reticulum, mitochondrion, MTX1, surface, April 2 S1, CD81, GPAA1, LGALS9, MGAT1, VAMP3, cell adhesion, ADAM15, beta-catenin, CNTN1 contactin, delta-catenin, channels and transporters, ABCG2 formerly BCRP1 xenobiotic transporter, CALM1 carmodulin grasps calcium ions, CALM2 carmodulin grasps calcium ions, HPCAL1 also grasps calcium. Upregulated in retina, GRIK5, MFSD10 also known as tetraenol tetracycline transporter-like protein, TFRC transferrin receptor, FOLR1 folate receptor, SLC25A11 mitochondrial oxogalactarate malate carrier, receptors, ACVRL1 TGF-beta receptor family Rindus-Osler-Weber syndrome, CD23AFCE2 low affinity IgE receptor, GRM4 metabotropic glutamate receptor, JAG1-NOTCH2 receptor associated with Alagille syndrome, MC2 melanocortin receptor type 2, SSTR5 somatostatin receptor tends to inhibit other hormones, LA cell recognition, BAT1, BSG basigen immunoglobulins superfamily, extracellular metalloproteinase, B2M beta-2 microglobulin, HLA-C, HLA-G, MIF macrophage migration inhibitory factor, TAPBP, kinase signaling, ADRBK1 can downregulate response to epinephrine, AGPAT1 acyl 3-phosphoglycerol acyl transferase, ARF1, ARF3, ARF4, ARF5, ARL2 or AS superfamily, CSF1 colony stimulating factor not highly expressed constitutively at 5 to 12, CSK-CSRC tyrosine kinase, DCT-duparchrome tautomerase, EFNA3, FKBP1A, GDI1 GDP dissociation inhibitor, GNAS1 ubiquitously expressed, but differentially imprinted, GNAI2, HAX1 associated with tyrosine kinases, ILK integrin linked kinase, MAPKAPK2, MAP2K2, MAP3K11, PITPNM phosphatidylinosytol. Transfer protein, RAC1-RuGTPase involved with many signaling pathways, RAP1-BGTPase involved with cell adhesion, RAGA-RAS-related GTP binding, STK19, STK24 serine 309 kinase, STK25, YWHA tyrosine 3 monoxygenase tryptophan 5 monoxygenase activation protein, beta polypeptide, YWHA tyrosine 3 monoxygenase tryptophan 5 monoxygenase activation protein, H polypeptide, YWHA tyrosine 3 monoxygenase tryptophan 5 monoxygenase activation protein, theta polypeptide, YWHA Z tyrosine 3 monoxygenase tryptophan 5 monoxygenase activation protein, zeta polypeptide, growth factors, AIF1, HDGF epitomer-derived growth factor, HGS, LTBP4, VEGFB, ZFP36L1, tissue necrosis factor. CD44 TNFRSF5, casein kinase, CSNK1E, CSNK2B, miscellaneous, ALAS1 aminoalevulinic acid synthase type 1, ARHGEF2 roguanine nucleotide exchange factor, ARMET mesencephalic astrocyte-derived neurotrophic factor, AES amino terminal enhancer of split, BECN1 involved in autophagy and partners with PI3K, BUD31 formerly maternal G10 transcript, creatine kinase CKB, cytidine demon is questionable, not present in very high levels at all, CPNE1, ENSA, FTH1 heavy chain of ferritin, GDI2 RAB-RAS vesicular trafficking, GUK1 guanylate kinase transfers phosphate from ATP to GMP, HPAT. Hypoxanthine guanine phosphoribosyl transferase A, IFITM1 induced by interferon, transmembrane protein, JTB jumping translocation breakpoint, MMPL2, NME2 nucleoside diphosphate kinase, NONO, P4HB, PRDX1 peroxyredoxin, PTMA prothymosin, RPA2 binds DNA during replication to keep it straightened out, SULT1A3 sulfate conjugation, SYNGR2 synaptogerin, tetratricopeptide, TTC1 small glutamine-rich, tetratricopeptide, open reading frame, C11ORF13, C14ORF2, C21ORF33, sperm testis. Although this page is devoted to genes that should be ubiquitously expressed, this section is for genes whose current name reflects their relative. Upregulation in testes, SPAG7, SRM spermidine synthase, TEGT-BAX1 inhibitor, DAZAP2 deleted in azoospermia, MEA1 male enhanced antigen, see also, inducible gene, references. http www.sagenet Index HTML, http www.tau.acl Housekeeping genes HTML, http www.tau.acl HKG/